The overall goal of multi-photon time-lapse imaging is to capture high-resolution, three-dimensional, real-time images of migratory cell populations.
This method can help answer key questions in developmental biology, such as mapping migration pathways of different cell populations. The main advantage of this technique over traditional confocal microscopy is that multi-photon lasers have deeper tissue penetration and decreased phototoxicity. This increases the usability in thicker tissues and allows for longer periods of image acquisition.
To set up for embryo collection, between 3 and 9 p.m., assemble breeding tanks and fill them with reverse osmosis or RO water. Transfer up to 3 females and 3 males into opposing sides of the divided tank. The next morning, remove the divider shortly after the lights turn on in the vivarium. Allow undisturbed mating for 20 minutes or until sufficient numbers of embryos are produced. The embryos will be at the bottom of the breeding tank. When eggs are observed, lift the slotted inner tank out along with the fish and quickly place them into a clean solid outer tank filled with RO water. Use an egg collecting screen to collect eggs from the outer tank and transfer the eggs to petri dishes containing 30 milliliters of 1x embryo medium. Incubate the collected eggs at 28.5 degrees Celsius. For transgenic embryos at 24 hours post-fertilization or HPF,